No? Okay. So question 1, this is about investigating the growth of different variety of yeast. Okay. Let me change color first and also thickness. So the growth of a variety of yeast. So what they do is that they say that the rate of respiration in a yeast is proportional to the biomass of the culture. So that means biomass would be the independent variable because they are relating this rate of respiration to biomass. So biomass is the independent variable here. So respiration rate can be used as a measure of the growth of yeast culture. So they use respiration rate to represent the growth rate, okay, of the yeast culture. So respiration rate can be measured using the redox indicator TTC. So this is where you need to be very familiar with redox indicator, right? So whether it is photosynthesis or respiration, there are a few ways to measure the rate of reaction. So in respiration, you learn that to measure the rate, the first method you learn is using respirometer, right, to measure the rate. You can also put the solution KOH or NaOH to absorb away the CO2. So whatever changes in the volume of gas is actually the volume of oxygen absorbed. And don't forget also, if you want to say soda lime, it has to be solid. So when the question asks you for a solution, you cannot write soda lime. You must write sodium chloride, sodium KOH or NaOH. So this one is aqueous, so solution form. Okay. So this is the differences you need to know. So using respirometer, okay. And a second way to measure respiration is, of course, we use a redox indicator. So redox indicator is actually a substitute for the NAD and FAD. NAD, N or FAD. So we know that in respiration, right, NAD and FAD, they will act as hydrogen carrier. So in the process of respiration, all the steps which involve dehydrogenation reaction, dehydrogenation reaction, it will lead to the removal of 2 hydrogen atoms, and these 2 hydrogen atoms will be carried by the NAD and FAD. So now when we use a redox indicator, the redox indicator will then pick up the 2 atoms from every dehydrogenation reaction in the respiration. So when they pick it up, the original is oxidized redox indicator, right? Okay, indicator has its own color. So when it pick up the hydrogen, it's considered as being reduced. Okay, reduced. It will become reduced redox indicator, reduced redox indicator, and it's got a different color. So because the redox indicator will give us a color when there's a reaction, so it's easier for us to measure. So instead of measuring gas like oxygen absorbed or CO2 release, right? If you want to calculate RQ value, you also need both oxygen and CO2. So in this case, we don't need to measure the gas. We're actually looking at time taken for color change, because the rate of reaction is either the rate whereby the color. So in this case, we're using what? TTC, right? So TTC is colorless. So this is very interesting. Is opposite from methylene blue and also DCPIP. So the 3 redox indicator that you may be usually used as a redox indicator is actually methylene blue, which behaves very similar to, it's not the same, DCPIP. Both of them behave in the same way. That means to say, original color, right? Oxidized version is blue, reduced version is colorless. So another one is actually TTC. So I'll use the opportunity of this particular exam question to actually teach you TTC. Because TTC came out in exam before in the past in A2 practical. Okay, when that time they're still hands-on, A2 practical. So TTC, oxidized version is actually colorless. And with this reduced version, it is actually a red color. So we got very little bit is pink color, but we got a lot of them becomes red color. So to measure the rate, we want to measure time taken for it could be, right, the rate of reaction, right? So you already know that there are two ways to measure the rate. Okay, the rate, either you fix the dP over dt, yeah? So the rate is dP over dt, right? D concentration of P over T. So you fix the time, so you put it at 5 minutes. So for 5 minutes, how much of the red color, means the reduced TTC, can be formed? Okay, so you fix the time, you measure the product. Or the other way around, you fix the product. Give me this one. Okay, you fix the product and then you measure the time. So by now you already know you must fix one and measure the other. So you can fix the product and then you measure the time. So measure the times means maybe you measure the time taken for the first red color to be seen, right? Measure time, the time taken for the first color to be visible. Then it becomes fixed. This one, measure the other. Now, if you want to fix the time and measure the other one, if you fix the time and then you measure the concentration of product form, right? In this case, the concentration is measured based on the intensity of red color. We need to use colorimeter. So I'm taking the opportunity to give you some more information about colorimeter. Okay. So you know that colorimeter, right, is an instrument where there's light put onto it. There's a light there. And then you need to use a filter. So this is something that I've not mentioned in class, right? So I think it's important for you to know, so I'm mentioning here. So you need to put a filter of certain color, and then your sample where you put your solution to measure the intensity of the red color. So this sample contains the reaction mixture, right? So this container is called the cuvette, C-U-V-E-T-T-E. This container is called cuvette. It's slightly smaller than the test tube. So this is where you place the solution, where you want to measure the intensity of the color into this cuvette. And then you switch on the light. So the light will pass through the your specimen, right? You'll pass through the specimen. Do I have an arrow here? No, we don't have. So the light will pass through the specimen this way, okay? So it'll pass through, and then it'll pass through. And then at the other side, you have got a sensor. So you got a sensor here, which can detect the intensity of light that has passes through it. So therefore, which means to say that in the cuvette, okay, so in the cuvette, so in the cuvette, okay, in the cuvette, when you have got a lot of the particles, right, the red color particles, the what reduced TTC, so these are the particles inside it. So what happens when you got a lot of particles is that when you shine the light through this reaction mixture in the cuvette, this particle will actually absorb the light. So therefore, the higher the concentration of whatever solution you want to measure, that means the more is the particles, okay? And then the more light will be absorbed. And therefore, when more light absorbs, means the absorbance will increase. So the higher the absorbance, which means to say that the lower is the transmission. That means the total light that is sensed by the sensor become less, meaning to say that when you got less light transmitted, you got more light being absorbed. Okay? So I hope you by now you actually know the principle of this colorimeter. So there's one additional thing I want to mention to you is now the filter. The filter, so you see, if my solution is red color, right? What is the color of the filter that I must use? Is it red color or other colors? Can you type your answer, please? Nurin? Can all of you type the answer? So I want to measure the intensity of the red color of this solution. So what is the color of filter I should put here? Because this is a possible exam question. They can actually ask you. So do I use red color or use any other color? Noreen? Okay, let me see what color you want to use. Red color. Okay, Elisa said red color. Okay, what about Tei Shun Sin? What color filter should I use? Red. Noreen said other color. Tei Shun Sin said red color. See, now I want the particle to absorb that color when it passes through it. So if you see me as red color, do I absorb red color or do I reflect red color? You see the particles are red color, right? So if the particles are red color, that means they will actually reflect red color. They will not absorb red color. But you want to measure absorbance, So can you use red color for the filter? Cannot, right? Because when you use red color for the filter, okay, this filter here, the red light will pass through, and when it comes to the particle will not absorb the light. It will reflect the light. So I want to use the filter where it gives the color that the red color particle absorb. So if it reflects red, I see red. So what is the color it will absorb? Red color again. No, right? So it may be blue color, something opposite to red, right? So probably this filter should be a blue color filter. Correct or not? So I should use a blue color filter. So give me the blue color filter. So therefore, I must put this as a blue color filter here. This must be blue color. Then when blue color comes here, the red particle will not absorb the blue. It will not reflect the blue. It will absorb the blue. Then it's correct because you want to measure absorbance to denote how many particles you have. The more particles, the more blue color absorbs. The less blue color light pass through. So the higher the absorbance, the lower the transmission. Okay, so this is something that today I think I'll take opportunity to actually give you more detail about this because I think they can actually ask you an exam, okay? Small thing like this. Right, so with that, you learn the principle already. Let's go to the question now. So you got a glass tube here, which maybe you allow oxygen to enter for the yeast to respire aerobically, right? And then you got a conical flask inside, you got yeast culture, containing yeast suspension and nutrient solution. So you want to culture the yeast, you want the yeast to grow. You must make sure that, of course, you must have the yeast, okay, because yeast give us the enzymes, all the enzymes for respiration. And then you also must have the glucose, okay? So glucose so that you can carry out respiration, so they can produce ATP for respiration for the cell to grow. So you have both of them inside that. So what is the question here? So they tell you that different masses of yeast were added because your independent variable is different mass of yeast. And then to a fixed volume will give you different concentration of yeast suspension. Okay? So your independent variable and therefore will be the mass of yeast. It can also be the concentration of yeast because they say put in fixed volume. Okay? So then each yeast suspension was added to separate flasks of nutrient containing glucose. So they say that all of them you put glucose, but they didn't say concentration and volume, right? So they didn't put it there, but they tell you is glucose. You have standardized them in terms of the substrate for respiration, which is glucose, but it didn't mention volume and concentration. Okay, so as you read the question, please underline the keyword so that it's easy for you to identify the keywords, the content of the experiment. Okay, a redox indicator was added to each flask, and the flasks were incubated at a constant temperature for a fixed time period. So, let me change the color. So standardized in terms of temperature is constant, standardized in terms of fixed period of time. So this is where some students make a mistake in answering the question. So I'm putting the yeast together with the what? The glucose, and in a certain temperature, in a constant temperature for a period of time. So maybe it's a 10 minutes period. It's not at a regular time interval. So then they thought I'll explain to you why some students, when they write certain kind of answer, is wrong. Okay, this is a period of time like 10 minutes. For example, it is not time interval. If time interval means maybe 2, 4, 6, 8, 10, every 2 minutes interval is called time interval. Okay, so that's the difference between a period of time versus time interval. So that is where some students make mistake in their answer and therefore didn't get the mark. Right, later on you'll see which question already. So next, the color of each suspension was modeled over the incubation period. So incubation period means the entire period of time, for example, 10 minutes. They didn't mention, right? They said a fixed period of time. So it's a fix. So we assume it's 10 minutes, okay? So we monitor over the incubation period. The keyword here comes here, over. Hello, where are you? Monitor over the incubation period using a colorimeter. That means to say that if the incubation period is 10 minutes, monitor over the incubation period means in between the 10 minutes, 2, 4, 6, 8, 10, probably I will read the red color at a fixed time interval over the 10 minutes of incubation period. This is the meaning of monitor over the incubation time period. Okay. So next, a colorimeter passes a beam of light through a colored filter, like just now we mentioned blue color, right, into a solution and measures the light absorbance of that solution. So that solution absorbs how much of the red light. So you're measuring absorbance. The more the particle, the more absorbed. So a standard solution is used to set the colorimeter scale to 0 before taking any measurements. So which means to say that originally when there are no red color, because the TTC was at the start where they are all oxidized version of TTC. So you use this as what? Colorimeter scale set to 0. 0 means zero absorbance. It means no light being absorbed, all light being transmitted, right? Zero absorbance. Okay? The original TTC solutions are all transparent. So it's 100% transmission of light. So 100% transmission of light means zero absorbance. Okay, 100% because no particle. So 0% absorbance is equals to 100%, what is it? Transmission, right? Transmission. So all the light transmitted through. So no particle. Okay. So next. So number one, state the independent variable and the dependent variable in this investigation. So independent variable is something that we deliberately change. So we already know that independent variable is, let's look at the mark scheme directly. So independent variable is, okay, I make it bigger so it's easier to see, okay? It is the biomass or the mass of the yeast, or because they also said fixed volume, so you make it into concentration of yeast. Okay, dependent variable is something that we measure, something that we are measuring from the experiment. So what do we measure? We actually measure by reading off from the colorimeter, the meter, right? The absorbance. So therefore the answer is the absorbance or the color change. This is where I want to bring to your attention. I have just, this is not sidetracked a bit, but this is something important here. Now, you look at your OneDrive folder, right? I have updated this principal examiner common mistakes. The one they've given to you in the past was 2022. Now, this one is 2024 because 2 weeks ago, I attended the Cambridge Professional Development and we obtained the latest 2024 examiner's key message to the teachers about common mistakes of the students. So different batch will have different common mistakes. So I have included the new common mistakes identified by examiner in 2024 to the existing 2022 document that are given to you. So everything compiled together inside here. So you see that some of the information there actually new one. So I specifically want to bring to attention, now before exam, please read through all these. What are common mistake for students in paper 4, okay, and then in paper 5, and then I want to bring to a specific attention to the way you describe observation in experiment, okay? This is very important just in case you are not aware of how important it is to write your answer in this way. Uh, Biochemical test is biomolecule here, right? So I want you to see that here, when there is no change, let's say you do a Benedict's test, when color is involved, and where there's no change in color, right? The observation, you cannot write no change in color, or, oh, no, sorry. You cannot write, or it's a negative result. You cannot write it's a negative result because examiner wants your observation to be what your eyes see, right? You cannot write negative result, then the examiner don't know what your eyes see. What is negative? So negative means what? So you need to write the color that you see. Even no change has no mark. You must tell me, let's say iodine test, right? So the iodine solution remains orange brown. So this is the meaning of negative result or no change. You cannot say no change. What your eye see? You see that after you add iodine solution, the iodine solution remains orange brown. Okay, so this is negative result. You must describe exactly what you see. Number two, the color that you use to describe, right? Actually, you can use two color. For example, orange, brown, right? But when you write the two color, because orange is one color, brown is one color, you must put a dash in between the two colors. This is the actual original color of iodine in the concentrated form, orange brown. But when you add to water, it's what? Yellow brown, right? Okay. And when you do the iodine test, when there's a presence of touch, the color is actually called the blue black. So please write blue black in this way. Examiner last time says you cannot write two color together. Blue is blue, black is black. You cannot write blue black, but actually the actual color is blue black. So if you want to write blue black, you must put a dash there. Okay, so this is where I want to bring to your attention because paper 5 also you need to write observation, right? So just in case observation is color, you need to write your color properly, color. Even like Benedict's test, right? You cannot say that, oh, a brick red precipitate is formed. You cannot write like this. You must tell me the color change from initial. Where's the color? The initial color, what is the color? And the final color is color. So the color change from clear blue to brick red precipitate. So you need to write the initial color, clear blue, okay, to brick red precipitate. You need to describe fully what your eyes see from beginning to the end. What is the first one? What is the last one? Of course, you don't need to tell me from clear blue change to what? Change to green, after that yellow, and then orange, and then brick red. So the first one and the final one. Okay? But it depends on scenario when you must read the question. Maybe sometimes you need to write the in-between. You need to read the question. Okay? So that's where I want to bring to attention for this one here. Looks like there's something missing here, which is the initial color and the final color. There should be one point inside here, but it's not here. So I mentioned to you now, okay? So let's move on to the question A. So my focus is more on, of course, when I discuss the paper, I also want you to learn something, right? That's why I don't do it very fast, right? Okay, someone got a question, no? So dependent variable is like the first result that you can directly measure and observe, not calculated, is it? Correct. Dependent variable is always something that you measure in the experiment. And then from what you measure, then only you calculate. For example, you want to find out what is the respiration rate. So you don't measure respiration rate, right? You measure the volume of oxygen released per unit, absorbed per unit time, right? You measure the distance traveled by manual fluid per unit time. This is what you measure. So this is your data. This is your dependent variable. Then from there, you do calculation. Let's say you convert rate to volume per time. So you must convert the distance measured to volume by pi r square, L divided by time. So that's called calculated. So the calculated is not the dependent variable. The dependent variable is what you measure. Okay, but sometimes you look at past year, they mix up. They accept both, right? Okay, but it is the one that you measure. Okay, in order to get to the final one that you want to know. But what you measure and then put the formula or equation to get the final one that you know, the one you measure is dependent variable. Okay, so let's go to the next question. Where am I? Here. So part 2, identify the variables that students have standardized in investigation. Oh, this one is very easy, right? Whatever they mention here that you can identify is standardized variable, they're all correct. So you can see that the standardized variable is the volume of water added, because they already said that it is fixed volume. Anything fixed is standardized. Okay? The volume of yeast suspension and hence is standardized. They use the same indicator, but they didn't say volume and concentration, right? As long as the indicator is same, it's also standardized in terms of that same type. You don't use TTC for this and use methylene blue for that and use DCPP for the other one, right? Because we're not comparing between the different kind of redox indicator. Okay. We use the same temperature, so standardized in terms of same temperature. Standardized in terms of same period of incubation, because they say fixed, okay? And then colorimeter has been zeroed. So we put the solution where TTC has not changed color, and we use this as a zero. So we already standardized the zero for all the temperatures that you want to read later on. Okay? After the reaction. So all these are standardized. So I really like this one. Colorimeter 0 is also standardized. You standardize the value for 0 is what color. Okay? You see, using colorimeter to measure the Benedict's test is more difficult, because Benedict's test, right, the color will change according to how much reducing sugar we have. It can be green, yellow, what is it? Green, yellow, orange, and red. So that means we have to use different filters, isn't it, to measure different absorbance for different colored particle. So it's more difficult to measure. Right? Have you ever thought of that? This is a possible question in exam. Right? What is the difficulty of measuring color change for Benedict's test versus color change using the redox indicator? Right? Okay. So today I give you a lot of tips. Okay. This is called exam tips. Right. So finish that one. Part 3, suggest a suitable control for this investigation. So every time enzyme is involved, right? The control, yes, you know, but do you know how to write to make sure you got the mark? It's very, very important here. See, in an enzyme catalyzed reaction, a control should be, we will replace. This is where a lot of students never say. We will replace the enzyme. So enzyme in this particular experiment is actually the yeast. So we need to replace the yeast by what? Boiled yeast, dead yeast, inactivated yeast. So usually most of you will say boiled yeast. Okay, of the same mass, right? You must standardize everything other than one is boiled, one is not boiled. The rest must be exactly the same. So the same mass, same volume. Now, if you say replace it with distilled water, it must be same volume of distilled water. So you can. Some student wrote distilled water, I accept. But you must say same volume of distilled water. Okay? Other than that, I don't want to use yeast, can or not? Can. I use something else to replace the yeast that is also solid. Because yeast is a solid inside the liquid. When you suspend it, that's why it blocks the light. If it's soluble, it will not block the light. Okay, it becomes a clear colored solution like copper sulfate solution. Copper sulfate dissolve, but it gives you blue color because of copper 2 plus. So it gives you clear blue color. It dissolves. This one doesn't dissolve, that's why it blocks the light. Okay, so it becomes particle inside. So I don't want to use this. I can use something that's solid. I can use glass beads. But this glass beads must have the same mass. It must have the same mass as the yeast. Very, very strange. Okay. And sterile. Sterile glass bead. Sterile means this glass bead must not contain fungus, must not contain bacteria in it, because then they start to use the glucose, and then suddenly you see that there's respiration. Okay, so it must be sterile, means maybe you put alcohol to sterilize the glass bead. This is a very good thing, because it can be contaminated, right, by fungus, right, by bacteria from the air. So sterilize it so that it will not cause respiration. So Therefore, remember to write, replace the yeast or replace the enzyme with a denatured enzyme, boiled enzyme. Boiled means you denature it. Okay? So this A part 3. So next question is A part 4. A part 4 is what? So you are asked to do something to the sketch. Sketch a graph to show the expected change in absorbance over time during incubation of yeast and label the axis. So very easy, right? So the axis is, what is it? Temperature, isn't it? Your independent variable is temperature. And then this is absorbance. So absorbance is as the TTC, accept this oxidized TTC as it accepts the hydrogen atoms from the dehydrogenation reaction of intermediate substrate of respiration, it will be reduced, and the reduced TTC is red color. So it becomes red particle, right? So we will expect as the reaction continues, the more product will be formed. So reduced TTC becomes a product. And this product is becoming more and more. So when you got more and more product, it will absorb more light. So when you got more product, and therefore you have got more red particles, and therefore more light will be absorbed, and therefore be more absorbance. So absorbance will increase. So most of the students get this correct, but some students got it wrong, right? So absorbance increase, how do I draw it? I draw the box. How do I draw it? How do I draw it? Draw it. Okay, like this. So this is something like product form with time like this. Doesn't go down, sorry, go straight, becomes horizontal. Okay? So that is the answer. Let's look at answer for A is what? Answer for A is, so x-axis is time, or you can write short form T, y-axis absorbance. I'm so surprised. Absorbance short form is AB. AB. So this is where some students don't get the mark because the time is not incubation time. Sorry. The time is not incubation period. The time is incubation time. How long the yeast has been incubated in that particular temperature? Correct or not? Correct, how long it has been incubated in that temperature? Because for the incubation period is 10 minutes. Every 2 minutes or every 1 minute, we read the absorbance. So therefore, the x-axis should be incubation time. How long it has been incubated before I measure. It is not incubation period. Incubation period is the whole period, 10 minutes, for example. That's why some students didn't get the mark for the axis because the student label as incubation period. This is where I want to stress just now that you read carefully. There's a difference between incubation period and incubation time. So this minor detail that now that you know your content very well, you want to brush up your sharpness of using the words. Different word is different meaning. Use the correct word. You must use the correct word for the correct thing that you want to describe. Okay? Blah, 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 blah. Okay, next. So do you know the difference between sketch and plot, isn't it? Plot means you got the points, the result from the experiment, and then you put it, the points onto the graph and plot it. Sketch means you use your theory, how the shape should look like, and then you sketch. You have no result. It's from theory you sketch. Okay, so the difference between sketch and plot. So next question B. So you need to describe a method that the student could use to compare the respiration rate of the 3 variety of yeast. So there are 3 variety of yeast that the student want to use to compare which one of the higher growth rate, and they are compressed yeast. So that's all. So you need to use TTC as redox indicator. So you must set up your method in a logical order. This one you want to pay attention now. When you want to write your method, it's just like how you did paper 3. when you need to carry out experiment by following the instructions given step 1, step 2, step 3. When you write your answer in this design experiment, you need to write it in a logical order. Okay, logical order means step 1, this is what you need to do first, followed by step 2, followed by step 3. Even though, of course, the easiest way for you to write is, of course, with the subtitle, number 1, independent variable, number 2, dependent variable. So number 1 is IV, right? Independent variable followed by dependent variable, and then standardized variable, and then the detailed procedure, and then the what is called reliability, okay, and safety precaution. Correct? So this is the best way for you to actually think of the points to write, but sometimes you need to rearrange them according to which one comes first. Okay, which one comes first? Especially when the question says in logical order. This is something that I think is important to know. Right. So here, so that means they give you what? Compressed yeast, you know. So you must start from compressed yeast and then start to your description. Compressed yeast. Okay, that means you need to make the yeast suspension, right? Out of 3 of them. And then TTC, what about the standardize the volume and concentration of TTC? What about temperature? So what temperature are you going to use? And then what about how long do you put them in that temperature before you compare the rate of reaction? So how do you get the rate of reaction, your dependent variable? Do you fix the time and then you read the absorbance, or you wait for the time for the first red color to form? Which one is better? It's actually the better way is to fix the time. Fix the time, because sometimes after 10 minutes, no red color form, you can stop experiment already. If you say that time taken for the first red color to appear, when there's no red color, you wait forever, so it's a waste of time. So it's best is to fix time and measure the other. Even the other way now is can. Okay, someone got question just now. Now no more already. Got question. Okay. Miss, can you repeat what are the 6 aspects to consider for design experiment question Teh Shun Sin? Can. This is basic, right? This basic I teach you last time already. Okay, since you're in the class, you request what I give you. Let me pull this thing here first. Okay. You know, I'm having problem to maneuver this thing here. Okay, clear all. So every time you design experiment, right, there's a subtopic that you can write. First is called the independent variable IV. I use short form like easier, IV. Then number 2, what is your dependent variable? So what do you want to measure here? Like just now I said, you can fix the time and measure the first color, measure the color change after the time, or you fix the color and measure the time. Means what is the time taken for the first color to appear, correct or not? So DV means what you measure, how you measure, and then the standardized variable, everything that you need to standardize, like temperature, like volume, like concentration, okay, like mass, etc. And then sometimes, like this one don't have pH. Sometimes when your substrate or product involves acid or base, the pH will change. Then you have to standardize your pH, and maybe standardize the timing of something. Okay, time to equilibrate temperature, time to equilibrate pressure, all this must standardize. Then after that, number 4 is what? Reliability. Reliability is like, remember, you repeat experiment to get 2 more readings. That means altogether total 3 readings. And after that, you have to calculate mean, right? So this is to increase reliability. Or from the repeats, you can ignore the error. So you identify the error or the anomaly. Sorry, anomaly is the word. So you identify the anomaly and you don't calculate the mean using the anomaly. Don't use the anomaly to calculate mean. Okay, reliability. And then number 5 is called what? The safety precaution. So you've done a lot of past here, you know that safety, you need to mention 3 things. Number 1 is hazard. So hazard means what is the object? What is the solution? What is it inside the experiment that cause hazard? Maybe fire that we use, electricity that we use, the water bath that we use, the acid that we use. Okay, hazard. So hazard, after that, risk. So this thing that we use, what kind of risk it can cause? Let's say iodine, it may stain your clothes. Iodine may cause irritation to your skin. So the risk is it may cause, so iodine solution is the hazard. Risk is what? It can cause irritation. Okay. And then what is your steps for precaution? So these three things must be mentioned for safety. These three things. So hazard, risk, precaution. Then only you can get the mark. You cannot say that, oh, there's electricity, so be careful. Then you cannot get mark. Be careful of what? Okay. Oh, using knife, be careful. No mark. Knife is a hazard. Risk is be careful. You can cut your finger. So it's precaution. Don't wear glove. Knife can cut glove. Cut away from the finger. Don't cut towards the finger, right? Okay. So these are the three things you need to mention to qualify you for 1 mark for safety. Usually only 1 mark for safety. Don't write 5 things there. 5 things also is 1 mark only. Okay. So 5 things here. So let's continue with the answer. Okay. Cannot. So let's look at the answer for design experiment, right? So I have categorized the answers into different categories. So it's easy for us to identify them and know how to write your points to score full marks. Right? When you categorize them to subtopic, right, you know that minimum you can score 5 marks already because it's got 5 different subtopics, and some of them you can write more. So that will give you like 6, 7, 8 marks already. Okay, so the first one is standardized variable. So because we are using yeast, we are using 3 different type of yeast, right? So the independent variable is the type of compressed yeast. So other than that, other thing about yeast must be standardized. What is it? Mass and volume that we use to make suspension from yeast must be the same. So this is called standardized variable. Even though the type of yeast is the independent variable, but the mass of and the volume of yeast is a standardized variable. Okay, you see the difference, right? The independent variable is the type of yeast, but we standardize the mass and volume for the 3 types of yeast. They're all the same, so they're standardized. Then number 2, concentration of glucose. So you must make the concentration of glucose the same, and the volume of the glucose is the same. And then so by now you know that you cannot just tell me you need to use this temperature. You need to maintain this temperature. You must tell me how to do. Everything that you want to design experiment, you must tell how to do. So maintain the temperature at optimum temperature can be any temperature between 15 to 80. Right? Because we do not know the yeast optimum temperature is how much, not our body and not 37 degrees Celsius, not human. So it can be 25, it can be 35, it can be 40, 45, you don't know. So safest one is below 40 and 25 can, 30 can, 35, any number between this range. So use thermostatic control water bath to maintain at this temperature. And be very careful that by now you know that whenever you want to use a water bath that is 0 degrees Celsius, okay, all to the way to 25 degrees Celsius or maybe 20 degrees Celsius, we cannot use thermostatic controlled water bath because the water at room temperature, depending on aircon, 21 degrees Celsius is already 21 degrees Celsius. How can you heat up 21 degrees Celsius to reach 0 degrees Celsius or 5 or 10 or 15, 20? Cannot. So we cannot use thermostatic controlled water bath when you want to use this temperature. You have to add ice to the water at room temperature. So this is something that you really need to read all the values, all the word in the question, and think out of the box to write your answer to score the marks. This is where the question will try to catch the student. So you have to be very alert and fully awake and energetic during the exam time. So you need to make sure that you sleep early, you drink the gaijing or whether aching, whatever, to make yourself alert, or maybe coffee, I don't know. Okay, and make sure you rest well and prepare well. Okay. Number 5, suitable temperature. This I mentioned just now already, right? Okay, so these are all standardized variables. See, standardized variable already 5 marks, already 5 marks already. So good, right? So procedure, remember not we are going to put the yeast in a water bath, right? So therefore, every time we put something to water bath, we need to allow time to equilibrate. So you remember before that last time when you do an experiment where it involves enzyme and substrate, and we want to test the effect of temperature, right? Okay. So let's say I want to use a temperature of 20, 30, 40, 50, 60 degrees Celsius, okay, temperature. I want to test the effect of temperature on the enzyme activity, and I'm working on enzyme that comes from yeast. So you learned before that in the water bath, you have to put the enzyme and the so this is my test tube, okay? You must put the enzyme and the substrate separately in two different test tubes, and to bring their temperature up to the temperature of water bath before you transfer the enzyme to the substrate. So this is enzyme. So you're transferring enzyme to the substrate. And this is enzyme. So you must heat them separately in water bath for at least 5 minutes to ensure that both of them are in the temperature that you want to test. Then only you transfer the enzyme to substrate. Because if you straight away take from the room temperature and then add them together and put them into the water bath, it will take some time before their temperature is raised to the temperature of water bath. That means we are creating error in our experiment already. We didn't run the experiment at the temperature that we want to test. So therefore, the result will be, the rate will be lower than the temperature that you want to test. If the temperature you use is higher than room temperature, that's why this is something that you need to know. That's why you see the marking point says what? We need to bring the yeast suspension and the nutrient solution to the temperature before we mix them. So allow time for the temperature of the yeast, which contain the enzyme and nutrient, to equilibrate with what? The temperature of the water bath before we mix these two solutions together. This is 1 mark. This is procedure. Then number 7, then don't forget to add TTC to the yeast and the nutrient mixture. Do I add the TTC later? I don't like to add TTC later, right? Okay, because TTC at room temperature can affect the temperature of these two. So probably I also get another tube which contain TTC is even better. Then add all the 3 together. This one very good. This one is reduced error in experiment. Okay. Next, dependent variable. Like that, very charm. Question 1 not yet finished. Dependent variable is what? Time for recording absorbance. So that means we are, what do you mean by time for recording absorbance? Time. So that means, let's say you fix, okay, what is the time taken for absorbent to be 20%? So you fix the absorbent at 20% and then you read the time. So what is the time taken for 20% absorbance to be produced? So every 1 minute, probably every 1 minute, you take out the solution reaction mixture, put it into the colorimeter and read the absorbance until you get a 20%, then that is the time that you want. So every 1 minute you take the absorbance, and then when you reach 20%, then you take that time. So time for recording absorbance, you fix your absorbance, read the time, or so that means you have to read the absorbance at regular time interval, right, to reach that 20%. Or you can leave it at a set time. After the time, you read the absorbance. So 2 ways to measure. So this is your independent variable. Okay. Then some more got standardized variable, right? So because we're using yeast, you know that when you use yeast, right, the yeast, because it's solid, it will settle down in the solution very fast. So every time you want to transfer the yeast, you must mix them properly, right? Mix them properly. And then when you add the yeast into the glucose, you have to mix them, right? Or else all of them settle down, right? Then some of the yeast will be blocked by another yeast. So you reduce surface area exposed to the substrate. So that will actually affect the rate of reaction. So you want to mix them throughout the experiment. Okay? Okay, give you more tips, Give you more tips. Today give you a lot of tips, You see, let's say you got 5 test tubes at one time. You cannot mix them, right? You don't have 5 hands to hold 5 glass rods to mix at the same time. Correct? So how are you going to mix them? My goodness. So there's this called stirrer. In experiment, we got this stirrer. So what is a stirrer? Stirrer is actually an instrument, right? With a metal in it, and this metal can rotate one. Okay, and then in your beaker, okay, of the yeast suspension, when you want to make it homogeneous all the time throughout the experiment, you will put a magnet stirrer into it. Okay, magnet stirrer is a very tiny little like a bullet, okay, that has magnet in it. So when you turn on the stirrer, so this machine is called the stirrer, this metal will start to turn. So when the metal turn, it will cause the magnet to turn. So when the magnet turn, it will stir this solution. So this solution will be stirred all the time, and the speed can be changed by changing the speed of this metal turning. So this is how we actually stir the solution when you got a few solutions that you want to stir, or you want to stir the solution for a long period of time. We don't use the stirrer. We don't use the glass rod. We use a stirrer. Okay, like this. Okay, so this to let you know another instrument that we use in experiment. Okay. So because we want the yeast to carry out aerobic respiration, according to the question, you must tell me what is the method to maintain oxygen concentration. How? You see, the question is very interesting because they draw the apparatus for you. Got or not? Got apparatus or not? Did they draw? Got got. You see, they actually put a glass tube here. So why they want to put a glass tube here? That means you want aerobic respiration means you must aerate this conical flask, right? How do you aerate this conical flask? Did they mention how? They didn't mention how, right? Mm. If you keep it open, cannot. If you keep it open, that means the air outside can come in, right? Okay. So keep it open for the air to come in. Of course, this is not so good, right? Second way is you don't stopper it. You put this conical flask onto the stirrer, and you put that sterilized magnet so that it will continue to stir this solution. So when it stirs this solution, it creates a current. So that current will actually bring in the air into the solution. So this is how you aerate it. This is how you aerate a solution, by stirring the solution. Okay. Another way is, this is the best way. Another way is you have to put this tube, right, all the way into the solution. And you must have got another tube that is here. Why? Because now you can actually pump in the air, right? From the outside, you pump in the air. So you pump in the air like how you pump the bubble into your aquarium, right? So you use a pump. So it will suck the air to this tube and goes into here, and it start to bubble. So that's where you get in the air which contain oxygen right into here. Then you must put another tube, because when you pump in the air, the pressure is going to increase. You must have an outlet pipe for the excess air to go out. So you got the air that comes in and the air that comes out, and you make sure you don't pump in the bacteria, virus, fungus from the air, because then they'll fight with the yeast for the glucose. So you must pass it through a filter that filter away all the microorganisms, the bacteria. So this filter must have got a pore size smaller than the size of the bacteria, right? So what is the size of bacteria? 0 point something, right? 0.2. So maybe you want to use a filter with the size of pore size of 0.1 micrometer, so that the air that you pump in is sterile. So you use a pump to pump the air, and then it must pass through the filter of pore size 0.1 At home we use 0.5 micrometer, right? Okay, so 0.1 micrometer, and then like this go out and come out. Okay, so this is something additional that I want to show you today here so that you can learn. Okay, so this is not like discussing your paper paper 5 only. This is like learning a lot of things, right, from discussion of this paper. So let's look at the marking continue. So come to number 11. So we must use at least 3 replicates. What do you mean by use? We must obtain, isn't it? Obtain 3 replicates. Actually, obtain 3 readings. Repeat experiment to obtain 3 readings. And that means some student didn't get marks. Right? They say, oh, repeat experiment 3 times. But then they didn't say calculate mean. Because of that, a lot of students, 1/3 of the students didn't get marked because of this. Must write after you get your replicates, what you do with it. You must calculate mean. Okay, so that from the 3 readings, you can eliminate any possible anomalies before you calculate the mean that will increase reliability of the result. Okay, so I would prefer you to write repeat experiment to obtain 3 readings to calculate the mean so as to increase reliability of the result. Okay, like this. Then safety is, you already said, right? You must name a hazard, state the risk, and then state the precaution. For example, yeast can cause allergy. So anything that's biological, any sample that's biological may cause allergy to some people. So you must wear glove, wear mask, wear goggles. So you don't come into contact with your hand, your face, your eyes. TTC is an irritant. So all the redox indicators, methylene blue, DCPIP, even iodine solution, they're all in irritants. So you must wear glove, wear mask, wear goggles. Okay? So that's all for today.